Alright, so today I'm going to be discussing a new system for detecting nontuberculous mycobacterial glycopeptidolipids using electrochemical DNA biosensors. My name is Dylan Poch, and my advisor is Dr. Andrew Bonham, and I'm very excited to be sharing some of our latest findings. So first off, what are NTM, or nontuberculous mycobacteria? There's estimated to be 200 species of NTM, and many of these are classified as opportunistic pathogens, meaning they typically will not infect or cause disease with someone unless that person is already immunocompromised or has a pre-existing medical condition. There's an estimated 86,000 people within the United States alone with NTM pulmonary disease, and a scary statistic is seeing that it's rising at about 8% each and every year. This is quite frightening, given that many of these NTM bacteria are growing antibiotic resistance as well. So what's the diagnostics for NTM disease? So currently it's culture-based methods. And this can take up to 20 months between the first onset of symptoms and the final clinical diagnostic. This is unacceptable, given that many of these people are immunocompromised and might not have 20 months to be diagnosed before treatment. So what about point-of-care or POC diagnostic alternatives that could bring this up to 20-month period down to a matter of minutes? So some examples of POC diagnostics that we all know would be the blood glucometer, a pregnancy test, or the latest one being the COVID-19 test. And all of these have in common, they're mass producible, they're able to be shipped directly to the patient's house, and you can get results within minutes without needing a clinical lab or highly trained staff to do so. We're putting that diagnostic power in the hands of the patient. So one type of point of care diagnostic is the biosensor, and there's many different types of biosensors, but all biosensors have at least these 5 constituent elements. So first we have a target. This is what we're trying to identify. In our case, we're looking at molecules on the outer cell wall of NTM bacteria. Then we have some type of biocomponent. So we're using single-stranded DNA aptamers, and an aptamer is just a nucleic acid that binds to our given target. You can use antibodies, enzymes, lots of different things. This binding interaction causes some type of change in the signal. Our case, that's electrochemical. In others, it could be optical, a pH change, even heat-related. And then some type of detector, we use a potentiostat, detects that change from the binding interaction, and we use analysis, square wave voltammetry, to actually quantify that level of the target in solution. So before going on to the specifics of our DNA biosensor, I wanted to introduce some of our previous work that we've done before. So originally, we're looking at this mannose-capped liporabinomannan, or MannLam biosensor, and MannLam is found on both NTM as well as tuberculosis, or TB. And we basically just pre-ordered a DNA piece that was allegedly specific to MannLam. But what we found was we had a lot of reproducibility issues with this, as can be seen here. So the y-axis is a change in the current, and then the x-axis is increasing concentration of that MannLam. And we did see a very significant change, but those error bars are very large and very inconsistent. So we decided to turn our attention to these glycopeptidolipids. And in my opinion, glycopeptidolipids are a much better target because it's NTM specific, so it's not found in tuberculosis. Okay, so getting more specifics with our biosensor, again, that target is that glycopeptidolipid, GPL. Then we've got that biocomponent, which is that single-stranded DNA aptamer represented by that blue squiggly line. And then we append that DNA onto a gold surface electrode. So the DNA is up here, and then we attach a methylene blue redox moiety onto that DNA. So methylene blue is simply a molecule that's very electrical in nature. So it's got a lot of electricity, and what happens is when we introduce GPL into the solution, it causes a binding interaction with that DNA. So that DNA undergoes a conformational rearrangement to bind to the GPL, and that causes that methylene blue electrical molecule to get closer to that gold electrode, or even further away depending on the system, causing a direct electrical current difference that we can measure, and based off of that, we can extrapolate out the concentration of GPL to detect if a patient had NTM or not. And here's just some photos of the electrodes that we use, as well as the newer rod electrodes that we've been using recently. So the first step is purifying that GPL molecule that we're targeting. So we got some bacterial cell cultures from our collaborators at National Jewish Health by Dr. Jennifer Honda. She turned those into bacterial cell pellets, and once I received those, I created total lipid extracts, which is exactly what it sounds like, extracting the lipid components. After I made those, we used a technique called thin layer chromatography, or TLC, and what you see here on this red box is the putative GPL, so that's where all of the GPL molecules kind of separated out of those total lipid extracts. So we essentially just scraped that band in the red box to get the GPLs, and then used microsilica column chromatography to purify it. And what you see in that test tube is our allegedly pure GPLs. So quite a process. It took about 1.5 years to do. But once we did that, we actually had to design this DNA aptamer. And to do that, we used a technique called the systematic evolution of ligands by exponential enrichment, or SELECT. And SELECT is a multi-step iterative process where we start with about 1 million billion nucleotide sequences. A nucleotide is just the constituent elements of a nucleic acid, like DNA or RNA. And what happens is, so in this test tube with tons and tons of nucleotides, we introduce that purified GPL molecule. And randomly, some of those DNAs will bind to GPL. And in step 2, we use this magnetic pull-down system where those DNAs that are bound get pulled down in solution. And only the non-high affinity DNAs are in the top of the solution, so we just kind of rinse off the ones that didn't bind. We don't care about those. And then we use polymerase chain reaction to amplify those highly specific DNAs. And what we end up with after about 8 to 10 iterations of this is very, very highly specific and highly selective DNA aptamers that bind selectively to GPL. So after using selects, we got this sequence shown above, and that is our DNA piece that binds to GPL. So then we use thermodynamical analysis to actually try to determine the conformations of our DNA aptamer. So what you see on your left is 3 different conformations of the exact same sequence of DNA. And those blue dots represent where the methylene blue molecule is appended to. And what we see is those delta G values, which is just a term we use for quantifying the energetics of the system, are all very similar. So these 3 conformations would exist in equilibrium, meaning they exist in roughly the same concentrations in a solution without GPL. But when we add GPL, essentially only one of those 3 conformations will predominantly exist, causing the overall electrical current to be different. Because before we were taking an average of those 3 positions, and now we're just taking one of those positions, so the current will be different. And then over here on the right is a tertiary, or kind of like 3-dimensional structure of, it's actually an RNA analog, but it should be very similar to how our DNA looks in kind of 3 dimensions. So now that we have that DNA sequence, we kind of know how it's going to be looking in 3 dimensions. We can determine if it actually binds to GPL as we assumed it does. To do this, we use a technique called fluorescence spectroscopy, and we attached fluorescein molecules onto that DNA. And fluorescein emits fluorescence that goes on to hit a detector called a fluorimeter. And our theory was, if GPL is binding with high affinity, those GPL molecules are going to kind of form a wall around the DNA and surround it, and the GPL will absorb that fluorescence, causing a decrease in the fluorescence. And that's exactly what we saw. So you're looking at a change in the magnitude of fluorescence with an increase in the target. And the blue curve is the GPL, so as we're increasing GPL, we see a huge change in that fluorescence as expected. And then we did some negative controls, and those had no significant change. So finally, we know it's binding, and I'm very excited to share this result because I actually just collected it on Monday, so it's very fresh, but we're able to actually test if there is a current change. And what we saw is there is. So y-axis, you're looking at the change in electrochemical current. X-axis, again, just that increase in the concentration of GPL. And it seems to be right now a signal off biosensor. So if my arm is that gold electrode and my finger is the methylene blue, it seems to be moving away from that electrode right now, causing that electrochemical current to decrease. But if we change the frequency of the square wave voltammetry, then we can actually create a signal on biosensor in theory, so we're not too worried. We're just looking for some type of change. But we wanted to make sure it's not just non-specifically binding to any molecule. So we did a negative control and with bovine serum albumin, or BSA, and we're very happy to say the BSA had no significant change. So what you're looking at now is a % change in the magnitude of that current. And we saw a pretty decent change, about 80% with GPL, but less than 5% with BSA, and we actually used 100 times the BSA molecules. So this tells us our sensor seems to be very selective and specific. Again, this is one test, so we need to make sure it's reproducible before we can draw any definite conclusions. So next steps, while this is all great, if we can take this to a clinic, that would be wonderful. But kind of thinking about the big picture, I think developing not only an NTM sensor, but an NTM and tuberculosis dual biosensor would be wonderful, because up to 20% of tuberculosis infections are actually attributed to NTM infections, or misdiagnosed. Additionally, many of the treatments for TB are completely ineffective for NTM. So if we're able to detect both GPL, which would detect NTM, as well as that original target, MANLAM, which can detect TB, we're able to differentiate between the two of those. So using this system, what you see is that gold surface electrode horizontally. We attach that same DNA aptamer with methylene blue, exact same stuff over here. And then we append onto a second redox molecule, so a second electrical molecule called anthraquinone. And this kind of acts as a baseline signal to rule out some of the background noise that we had issues with in the first biosensor. And then additionally, we would attach a second DNA aptamer that's specific to MANLAM. And that would have ferrocene on it instead of methylene blue, so we're able to actually differentiate between those peak potentials. And basically, the system would allow us to take the ratio of the signal to background noise by, for instance, looking at the GPL-specific aptamer, looking at the change in the current from methylene blue to the change of your current to anthraquinone, and vice versa. And I really think this would make it a lot more consistent and reproducible, and allow diagnosis and differentiation, which right now is just impossible with recurrent diagnostics. So this project does have a lot of limitations. One, we've only done one trial, so we have to reproduce that. Two, GPL purification. So our method is crude at best. Right now we're saying, hey, this looks like a GPL band, so we're scraping those and hoping it's GPL. That's based off of the literature values of where the bands kind of lie on the TLC plates, but we can do a lot better, so we're hoping to use like LCMS to actually verify is that GPL, because there's some literature values on where GPL should be on a mass spec. Additionally, all of our tests have been in buffered solutions, so we actually have to now test in blood, sputum, and complex media, because that's what we would be looking at in the clinic if it ever goes to the clinic. So I just want to leave you with reducing this up to 20-month-long diagnostic period to diagnose NTM to a matter of minutes could greatly improve patient outcomes, because now we can start treating these patients on a much more efficient timeline. If we look at some of the leading causes of death by the CDC in 2020, we've got heart disease, COVID, and cancer. But using these types of point-of-care diagnostic systems that I've talked to you about today, and then complexing those with personalized interventions, such as CRISPR therapeutics, we can create custom pharmaceuticals on a time frame that has up until now been impossible. And I strongly believe using this type of system we can greatly reduce mortality rates associated with not only these diseases, but many biomedical problems we face today. Thank you. I did just want to acknowledge too and thank my advisor, Dr. Andrew Bonham, for just kind of teaching me all of this stuff for the past couple of years and being a great mentor, as well as MSU Denver, Dr. Jennifer Honda, my family, my girlfriend Irene, and just everyone who's supported me through this process. Thank you very much.